Um, well, it's a real pleasure to be down here and presenting. And I enjoy coming here. My parents live at a place called Dash Point, which is near Tacoma. So I stayed there last night. So I don't mind coming back. So I'm going to be telling you about mouse imaging experiments that we do. And our lab works on really two things. We work on stroke, so there's an infarct here in the mouse brain. But to really study stroke, we need to appreciate cortical connectivity relationships. And so we've been making strokes in different lines of mice that allow us to visualize connectivity, alter function, channel rhodopsin mice, and so on. And the level of organization that we're interested in is what's termed the mesoscale, or sort of middle scale. And this is not this microscopic synaptic scale or the whole brain, but what's in between. So these are projections within a structure, sort of between homotopic areas in the cortex, or between two related structures, such as the thalamus and the cortex. And we're really taking advantage of a lot of the Allen Institute resources to assess this mesoscale functional connectome. And basically, our interest is in what happens after you after there's a spontaneous or experimentally induced infarct in the mouse brain. And so, of course, some tissue is going to die, but there's lots of surviving circuitry. And sort of the story in the literature is that animals recover by transferring the function from what's infarcted to what survives. And I won't talk too much about this process of remapping, but I will go into some preparations for studying the mouse brain from this perspective. So sort of looking at it as if we're flying over the top of the mouse, and it's sort of a flattened perspective of cortical functional connectivity. And again, this is our perspective in this cartoon. And so we do a lot of wide field imaging. We also do two-photon imaging. But most of what I'll tell you about is wide field. And then looking at the brain from this flattened view, we don't try to resolve anything in the z-axis, in the cortical column. We assume that what we see at the surface is reflecting what's going on largely in the entire column. Although we do have some methods now with electrodes to get laminar information, but I won't talk about that. So we use we assume a sort of a columnar behavior. We actually have quite a large focal volume using these methods, about 1 millimeter. We use stable LEDs for excitation, and a variety of different preps. So initially we did this with organic voltage-sensitive dyes, such as RH1692, which give you great temporal resolution, but they're only good for acute experiments. Lately, we've been doing imaging through intact bone, and we really like this preparation because it's fairly non-invasive, and we use this with transgenic mice, and these are lines that we've actually gotten from the Allen Institute, and Hongkui Zheng made them available to us early on, the AI93, AI94, and others. So to assess cortical functional connectivity after stroke, we built a system where we could measure functional connections using light, using voltage-sensitive dye imaging, and at the same time stimulate channel rhodopsin. And the best combination when we started these experiments was to use channel rhodopsin for the photostimulated opsin, and then the organic voltage-sensitive dye, RH1692, to read out activity. And the idea is that with a laser, we can activate a particular cortical point, and then using the imaging, we can read out connection strength with that point. And we wanted to do this to look at how the brain changed after a stroke. And before I go into the data, I just want to introduce you to a number of very strong cortical consensus activity patterns that we can see in the mouse brain. And these activity patterns are also very similar to the major intracortical connections that you can see in the mouse connectivity atlas and the brain explorer from the Allen Institute. So if you take a lightly anesthetized mouse, you can also do this in a awake mouse. It's just easier under light anesthesia. And you give it some whisker stimulations and then average these responses. Now we're just looking at only the right hemisphere. And so this is over millimeters of cortex now. It's really, it's this whole area in this atlas view. So you stimulate the whisker, and you get a particular whisker pattern. And there's a little island up here. This is the motor cortex. And then over time, 25 milliseconds after moving the whisker, it spreads out into S2. If you give an auditory stimulus, you get another unique pattern. You get A1, and then you get a more medial area, which is this parietal association area. Forelimb stimulation, another unique pattern. Forelimb S1, and then that island out there is S2. And if you stimulate the hind limb, you get two little islands of activity in S2. I have no idea why, but they typically show up. And here's a visual stimulus. You can see the response spreads. This is a flash stimulus. And then you can see some activation in the cingulate cortex along the midline. So some higher transfer of visual information. So this is just a short video clip showing you the average forelimb response. You see a response in forelimb S1, and then you'll also see S2. And this type of response is easiest to see using the voltage-sensitive dye because you've got this high time resolution. We're certainly interested in also doing this with voltage-sensitive fluorescent proteins. Okay, so how is this related to optogenetics? So this is the forelimb stimulated response. So this is, on the top, is what you get when you stimulate the forelimb. And we did a sort of a naive experiment in the animal. So we said, okay, so now we're not going to stimulate the forelimb cortex, but we're going to stimulate forelimb S1 directly with a laser. And what you see is you get activation around the laser spot, but you can also pick up this little island of activation, which is exactly in the same place as S2 that you get when you naturally stimulate the forelimb. So the message here is if you photostimulate with optogenetics, so now we photostimulate S2 of the forelimb area, and we can get an activation independently in S1. So by photostimulating different points in the cortex, and this is using a Thai-1 channel rhodopsin mouse, Guopeng Feng made, we can get maps of cortical connectivity. Now, I'll skip this. This is just doing the same thing in the whisker system. And the reason we do this is we want to get a map of intracortical connection strength after stroke. So I'll skip ahead here so we can photostimulate a number of different regions of interest. These are visual areas, motor sensory areas. You can even see some areas that are hubs, parietal association area. You stimulate it, almost everything responds. So the ultimate goal is to make something like this, a connection map. So we've got regions of interest here in the cortex, parietal association area, hind limb, motor cortex. And then now the size of this symbol is proportional to the total in and out strength through that structure. And each arrow shows connection strength, and the thickness of the arrow corresponds to how strong this is. So we photostimulate a point, and then we measure the connection strength emanating from that photostimulated point. So our interest here was to make a map of both hemispheres to see what happens to the brain after stroke. So there's a phenomenon in the literature called diaskesis, which is a disconnection syndrome. So in the normal brain, this is from a beautiful review article by Tononi, you have interconnected networks. Everybody's okay with this. And after stroke, of course, you're going to get a big area where there's going to be a deficit. But what we need to understand is that this infarcted area, it's of course connected to everything else indirectly through these beautiful connections that the Allen Institute helps define. So we just asked a simple question. We make a connectivity diagram before stroke or in a sham stroke case. And so now this is both hemispheres, and the arrows correspond to connections between areas. And all you need to know here is we make a stroke on the right side of the brain. And so we use a light-activated method to make a very sort of defined stroke within the forelimb somatosensory area. So we knock that out and we confirm that histologically. We don't produce any damage in the other hemisphere, but what we see is if we just do a very simple color coding here, now we color code every connection that goes down by more than 15% red, and this is 1 week after stroke. What you see is there's a lot of loss of connectivity around the infarct, but the other hemisphere is also going down in connection strength. So these are off-target disconnection syndrome. So this is an animal model of diaskesis. Now, when the animal recovers, there's some spontaneous recovery between 1 and 8 weeks. And what you see is the recovery is supported not only around the infarct, but there's also a lot of increases in connection strength, now colored a gain of more than 15% is green in the other hemisphere. So when we think of psychiatric disease or injuries such as stroke, we shouldn't just think around the lesion. We need to think about the entire sort of the functional connectome. And another message is I think there's also opportunity in understanding some of these other other connections as they might provide routes for treatment. Okay, so how else can you get data from these animals? So I showed you photostimulation, sensory stimulation. We can also examine spontaneous or sort of resting state activity in the mouse. And we've done this with voltage-sensitive dyes, so very high time resolution. This is, again, light isoflurane anesthesia. Bregma's marked with a white dot. And you see these patterns of activity that emerge. And it's not like snow on a TV, but you see activations that are in the form of things that look like maps. And so you can analyze this using correlation, doing what we call a seed pixel correlation, taking a single pixel and then looking at how other pixels are correlated with that over a long sequence of time. And so what we've done here on the right is spontaneous activity. On the left is a movie of correlation, which updates every 100 milliseconds. And around this seed, which the laser pointer is pointing at, and you can see with just 5 seconds of data, we can get a remote connection here with a very high correlation back to the seed. And so the seed is in the S1, M1 area, and that's the secondary somatosensory cortex that we believe is showing up. So we can use correlation to define maps. I'll skip over this. So sensation, channel rhodopsin, spontaneous activity, all give us fairly similar maps. So can we do this in vivo, chronically? And this has been a big effort of the lab and really aided by these new tiger or tigri lines. The AI lines are wonderful. And we've been using these with an intact skull imaging method. And this is a very low-tech method for wide field imaging. All you need is a stable excitation source, an emission filter, and a camera, and you can collect images of brain activity. The hard part is figuring out what they mean, and we can talk about that later. But it's possible to image this macro level activity through the intact bone. So I'll skip ahead. So we've been doing this in the Allen Institute GCAM6 slow and fast lines, AI93, AI94 are some of them that we've worked with, and we've used EMX Cree driver to get expression in excitatory neurons. And we got these mice, I guess it was it's about 2 years ago now, and this is Matthew Vanny and Greg Selassie in the lab, and they're smiling here because we just got the new Allen Institute mouse. It was the right genotype, and the signals were so large that they thought that they could image it with their cell phone. They'd never seen anything like this. There's other mice we used, we had to do all kinds of image processing, but these Allen Institute mice were terrific. So there's a mouse under there, and there's a cell phone above it. It's awake, and that's an image on the Samsung phone. And I'm actually going somewhere scientific with this, you'll see. There's an iPhone here for monitoring the behavior. Now, we never published that. It's just deemed as fun, not science. But here's the phone. This is activity, spontaneous activity through the phone. And then now this is using statistics to pick out expected relationships between the barrel cortex and motor cortex. So we can get maps with a pretty low-tech sensor. So we were inspired by this and thought, maybe it's possible that instead of bringing these animals back to the lab and giving them anesthesia or trying to immobilize them, that we could perhaps bring the imaging to the mouse cage itself and to the animal facility. And we were really inspired by this paper from David Tank, which used rats and looked at 2-photon imaging. And they trained rats to self head fix, and they were doing cellular imaging in the rat visual cortex and also somatosensory cortex. So we thought, well, we want to try this with mice, and the system they have is a huge, Airtable and the full system. But we wanted something really compact that we could bring to the animal room. And this is Federico Bolanos, who's going to present a poster here. And he's a graduate student now, but he started this as an undergrad in his first year. And he got our lab working with these miniature computers, Raspberry Pi. It's kind of like an Arduino. And so we made mice with a head-fixing bar and one of these chronic windows. And we even used the camera that came with this. And these things are really dirt cheap, but very accurate. And we together with others in the lab, with Jeff Ledoux and Jamie Boyd, we built software and hardware for immobilizing the mouse voluntarily within its own cage. And these mice are water restricted. And then they come in, they get head fixed, and then there's the Raspberry Pi camera here. And so we can image the brain. You can also use it to photostimulate the brain and stimulate different areas. And one of the best things about it is we can keep 5 mice in a cage, and we use RFID tags to keep track of them. And so they come down a track They make electrical contact on these plates. That initiates some solenoids to move forward. The mouse is transiently immobilized, gets some water. We give it some sensory stimuli. We basically do a little experiment, image its brain, and then we release it. And then another mouse from the cage will come in, a few minutes later. So this is the mouse being imaged. This is the camera. It's a standard epifluorescent system. We have an RFID tag system where it's about the size of a grain of rice implanted under the skin. You can put these tags in with a syringe. It's very straightforward to do. You don't even have to do sutures. There is a metal bar here which is involved in attaching the mouse and the transcranial chronic window. I'll skip forward. So this is this is sped up, but just showing the mice in the cage, goes in, gets head fixed, and then it will come out. In this case, it's only being imaged for 30 seconds. It can be longer. It's going to come out. Let's see. There it goes. And then some other mice will look into it, possibly go in. So here's a different perspective, mouse getting head fixed and getting water, of course. So there can be, we've done some experiments where we have a screen out here and we can display horizontal or vertical bars. This mouse is being head fixed, and then it leaves and then comes back for a second session, which is quite common. So the mice, we of course track their weights and their health, and doing this, you can generate a lot of data, over 7,000 head-fixed imaging sessions in 90 days' time. This is from a total of 11 mice. And this is some of the data. We're still analyzing this. So this is video of the face and then brain activity simultaneously. And the closer we look at these mice, the more things we realize they're doing with their whiskers or sniffing. It's going to be very, I think a challenge classifying this. The tail moves a bit in the back. So now one thing we're doing is having multiple streams of video from the body, and you'll see the water reward here. And then this is the millimeter scale, both hemisphere imaging. So you can do this type of imaging here over 60 days approximately, and this is using correlation to define relationships between the barocortex and motor cortex are fairly stable. This is hind limb. This is a water reward response here in the anterior areas implicated in the tongue by Carl Swoboda and others. And these are visual responses. This is, I apologize, is fairly unsophisticated. It's a single yellow flash. And you see activation in the posterior part of the brain here. And these are 3 different mice. And very nice signal to noise. And this is more data from in the cage. So I better finish up here. So advantages of this method. So one of the things is it keeps the social structure in the cage intact. We don't have to disrupt their circadian rhythms. They keep their nest, all that. Of course, we can collect data 24 hours a day or photostimulate, inhibit. Another thing is the water is dispensed throughout the day instead of only during the training is potentially an advantage. And you could also use it in really challenging environments. If you wanted to make a germ-free mouse cage, microbiome research, hazardous pathogens, it's great for that. I'm going to skip this. Katalin from my lab who's also has worked here and is still involved in projects here is going to present something on using single-cell electrophysiology and correlating that with this macro scale imaging. But I'll leave that poster for him. So what I hope I got you to appreciate is that we need to think about this sort of this mesoscale perspective in the brain, and by understanding this connectivity, we may find some new opportunities for treating injury or psychiatric illnesses. So I think I'll skip to my acknowledgements. And so I'd like to thank all the people in the lab, in particular Federico and then Kat, and also Dirk, who's here, and our funding agencies. And thank you for the invitation.
Thank you, Dr. Murphy, for that really exciting talk. We have time for about maybe 2 questions. So there's some microphones circulating. If anyone has a question, please raise your hand.
Tim, you have these you have these beautiful maps of how connectivity changes about a week after a lesion. Do you have any sense of how that changes over time? Presumably that develops. And as you can imagine, I'm particularly interested in sort of acutely what happens when you inhibit.
Acutely, you mean in the like an hour after, or during? So one of the problems with using GCAMP6 in stroke is because it's so sensitive, there's a period just after the stroke where there's very high calcium levels. And there also are spreading ischemic depolarization. So it's been tricky using a chronic method like GCAMP. So we do have some data with voltage-sensitive dyes early on. And you can even see redistributions of function in the other hemisphere over short time scales.
Yeah, you have those data for whole brain imaging in response to sensory stimulus and spontaneous, both in I guess anesthetized and awake.
We've also done the same in awake.
How they different?
They're not
describe to us. Yeah.
So if you look at something like a sensory map, like the whisker map, it is fairly similar in both in the awake and anesthetized animal. And I think that part of this is that what we're seeing on this gross macro level really reflects these very strong consensus projections. But I think if we went in and we looked at, I don't know, power and particular frequency bands, things like that, we're going to see more subtle changes. But the gross macroscopic organization does not seem to change much in anesthesia. It's I think these are just a sort of invariant, hardwired connections.
Even the spontaneous activities.
And with spontaneous activity. So one limitation is how we've analyzed the data. So in a lot of cases, and here we've used correlation to produce these maps, and we're starting to find that correlation is a great way for showing you a map, but it's a difficult way to tease out very subtle differences in activity. I mean, I think what we're sort of mapping here is, it's like the equivalent of Route 5 or something in terms of, it's a highway, not something more subtle.
So I just have a quick follow-up on that, if I may. So you mentioned Tononi, and you may know that he's shown under anesthesia and natural sleep states that connectivity actually breaks down. So I'm wondering, when you do your photostimulation, have you seen differences yet in the awake brain, those latencies that you showed as you did with anesthesia?
Yeah, we've done that with light anesthesia. We have not. I mean, I should have brought more of our figures, but we we have in the things like the connection between the barrel S1 and M1, it is very similar in the awake case and under isoflurane or urethane anesthesia. So yeah, the data I showed you with the mouse where it was drinking water, there you see these large-scale patterns, and of course it's awake. It hasn't had any anesthesia for months. So there is, you know, we could talk more after, but the major connections seem to be intact.
Great. Well, thank you very much once again. I'd like to invite